Welcome to the stem cells module. In this talk, we're going to learn about adult stem cell niches. So the way we have embryonic stem cells, we also have stem cells present in adults. So after development is complete and the organism achieves adulthood, numerous organs will possess adult stem cells which are multipotent. These stem cells can produce a restricted array of cell types and can undergo a finite number of cell divisions. So as shown in this figure here, we have an example of a hematopoietic stem cell, and this hematopoietic stem cell is multipotent. So, when the multipotent stem cell divides asymmetrically, its maturing daughter cell often goes through a transition stage called the transit amplifying or progenitor cell. So in this figure, the hematopoietic stem cell, which is multipotent, can undergo cell division and give a myeloid progenitor cell, which is a type of a transit amplifying cell or a progenitor cell. Another name for these transit amplifying or progenitor cells are the precursor cells. Now when we look at this figure, the hematopoietic stem cell can give rise to either a lymphoid progenitor or a myeloid progenitor, and these can give rise to different types of cells that are found in blood. The hematopoietic stem cell is multipotent because it can only give rise to cells that are present in the blood. However, the hematopoietic stem cell cannot give rise to cells that are found, say, in the liver, and that is why it is multipotent and not pluripotent. We're now going to look at what happens in adult stem cells. So what we looked in the previous slide was in embryonic stem cells. Now, adult tissues and organs also have stem cells. Now, the properties of a cell that allows it to be an adult stem cell are that it must maintain long-term ability to divide. It must be able to produce differentiated daughter cells, and it also must be able to repopulate the stem cell pool. If a cell is able to satisfy these three conditions, it qualifies as an adult stem cell. The adult stem cell is housed in and controlled by its own adult stem cell niche. This niche provides all the cells and factors that are needed for regulating stem cell self-renewal, survival, and differentiation of the progeny into the appropriate cell fates. We're going to learn about the different stem cell niches that are found in different tissues. There are many different stem cell niches that are found in different organs of adult organisms, and some of them have been listed on this slide, but this list is not complete. So we do find stem cell niches in reproductive organs like the testes and ovaries, and these stem cells are used to form the male and the female gametes. We also find stem cell niches in the brain, intestines, bone marrow, as well as the mesenchyme. We can also find stem cells in skin too, and thus adult stem cells are quite abundant in an adult organism. We're now going to look at the adult intestinal stem cell niche. So in the epithelial lining of our intestines, there's the intestinal stem cell niche. And when we look at where it is present in the epithelial lining, it's actually present in the crypt, which is shown in the blue box in the figure. The stem cells are present at the base of the crypt, which is shown in the red box. So these are so-called the valleys, but when you look at the hills, which are the upper parts, that part is the villus or the villi, and the crypt is the lower valley. And right at the base of the crypt, we will find the intestinal stem cells. Now, the division of the stem cells in the crypt promotes the vertical cell displacement, and so the cells start moving towards the villus. This is shown by the arrow that we can see to the right. This is also shown in the form of the plane of cell division that is shown in the blue box. So the cells that are present at the base of the crypt, when they undergo cell division, we can see that the plane of cell division is a vertical plane. Okay. However, when we look at the cells that are upwards, they undergo cell division on the horizontal plane, and this promotes the vertical displacement of cells towards the villus. So it is at the crypt where we actually will have cell division or cell generation. So while cell generation occurs in the crypt, cell removal tends to occur in the tips of the villus that are shown in the blue box. So in this case, we can see that the rapid turnover that happens in epithelial's lining of the intestines is due to the removal of cells at the villus. And a lot of times the cells that get detached at the villus undergo cell death because of the process of anoikis. Now, there are a few regulatory mechanisms present in the intestinal stem cell niche, and in this case, let's look at how that happens. So, there are a few different types of stem cells that are present, but the most predominant intestinal stem cell are the crypt-based columnar cells, or CBCC. They are found along with another type of intestinal cell, which is not a stem cell, which are the Paneth cells. And that's what we can see in the figure in the blue box, where we see a CBCC cell, and right next to it is a Paneth cell. The intestinal stem cells are identified by expression of the protein LGR5. So if you want to know whether the cell that you're studying is an intestinal stem cell, then you should see whether it makes the protein LGR5. In addition to that protein, they also express NOTCH, Frizzled, and the bone morphogenetic protein receptor. These are all membrane proteins. Now, there are cells that are associated with the CBCC cells or in general with the crypt of the intestine, which are the stromal pericryptic cells. And we can see they're the cells that are in that box that I have made. Now, they regulate the stem cell niche by secreting molecules like Wnt2b and BMP4. And Wnt2b and BMP4 regulate stemness and differentiation. The Paneth cells also produce proteins like Wnt3a and delta-like 4, which will also promote stemness and differentiation. Wnt signaling promotes stemness, while BMP signaling promotes differentiation. Notch signaling, which is where the delta-like 4 plays a role, promotes sustained proliferation and lineage specification. So to remain a stem cell, the cells will respond to Wnt3a very well and to the Wnt2b because this Wnt signaling promotes stemness to allow them to retain being stem cells. On the other hand, BMP4, which is produced by those stromal pericryptic cells, it promotes the differentiation. Not signaling, which is sustained through delta-like 4 signaling, it promotes sustained proliferation and lineage specification, and hence non-signaling allows the cells to differentiate into the intestinal epithelial cells. Now, the LGR5 expressing intestinal stem cells can form all the differentiated intestinal epithelial cells. So that is quite remarkable because there are different types of epithelial cells in the intestinal lining. Experiments have been performed with confetti mice, and they show that a single CBCC stem cell can repopulate an entire crypt over time. And that is what is shown in this figure. So in these mice, the intestinal stem cells can be observed because they are expressing the LAC-Z gene, which produces beta-galactosidase. The beta-galactosidase can be induced to undergo a catalytic reaction to give a blue color. So when we look at these blue boxes where we see those blue dots, that is where we have the CBCC stem cells. Now what we can see is over time, so in the middle panel some time has passed, we can now see more cells showing blue coloration. These cells arose through the cell division of the CBCC stem cell and are undergoing differentiation to produce the intestinal cells. As we wait for some more time, now we can see that more than half of the entire villus is actually made from those CBCC stem cells because they appear blue. And thus, one single CBCC stem cell is able to repopulate an entire crypt over time. We can also see that in the lower panel of the figure, where the blue cells are the ones that are the CBCC stem cells or arose from the CBCC stem cells. The CBCC undergoes symmetrical cell division. So what that means is when the CBCC stem cell undergoes cell division, both cells that are formed are same in nature. So they form as the same type of cell. However, their location specifies whether they will be a stem cell or they are going to undergo differentiation. So the daughter cell that is away from the parent cell progresses through the transit amplifying progenitor fate. So in other words, when the CBCC undergoes cell division, the cell that is closer to the parent cell retains stem cell properties, whereas the daughter cell that is away from the parent cell, it starts becoming a transit amplifying progenitor cell and will move on to further parts of differentiation. Thus, the location of the daughter cell defines whether the cell will be a stem cell or it will undergo differentiation.
With this, we come to the end of our adult stem cell niches, where we learned about different stem cell niches that are found in adults and focused on the intestinal stem cell niche.